Buongiorno, sono qui per presentarvi la nuova edizione di Trieste Next. Il titolo della 14ª edizione di Trieste Next è La vita dentro, dialoghi tra scienze e tecnologie. 3 giorni per riflettere su come la scienza e la tecnologia ridefiniscono la vita, su come le discipline STEM dialogano con le scienze umane e su quali nuove domande emergono per il nostro futuro. Medicina di precisione, intelligenza artificiale, sostenibilità ambientale, robotica, biotecnologie e tecnologie quantistiche sono solo alcuni dei temi al centro di Trieste Next. È questo il fil rouge del festival, con un programma di oltre 100 eventi in inglese e italiano e oltre 300 relatori di fama internazionale. E ancora, il premio Trieste Next Science Book of the Year, le 70 attività per le scuole e i 40 spazi espositivi e laboratori aperti a tutti in Piazza Unità. Prima di accogliere gli ospiti del prossimo evento, a nome dei promotori e copromotori della manifestazione, vorrei ringraziare tutte le istituzioni scientifiche di Trieste, città della conoscenza, e le istituzioni che hanno collaborato con noi. Il main partner del festival, i partner e i content partner della manifestazione per il contributo fondamentale. Grazie anche ai media partner e ai partner tecnici. Infine, un grazie speciale ai 200 volontari di questa edizione. Ricordatevi anche di controllare il programma aggiornato degli eventi su www.triestenext.it, al quale potete accedere anche da questo codice QR. Buon ascolto e buon festival!
Can you hear me? Yeah. Thank you for joining us today at this event, Discovering Life Sciences Through the AI Lens. I'm really honored to be here today. We will cover many difficult and complex topics, from therapeutic advances to AI tools to decode the DNA and the genome language. So I would like to introduce you Alberto Cazzaniga, Dr. Cazzaniga here, researcher, head of Lab of Data Engineering at the Area Science Park. Thank you for joining us today. I would like to introduce you also Dr. Francesco Iorio, group leader at the Center of Research in Computational Biology at the Human Technopole in Milano. Thank you for being here today. And Alona Sosinsky, Dr. Sosinsky is the scientific director of Cancer Genomics England. Thank you. It's a pleasure to have you here today. So let's start with you, Dr. Sosinsky. Just a couple of information. There will be a Q&A session at the end. So at that point, you can feel free to ask many questions you may have. People joining us online cannot ask any question. But I know that our guest will be more than free to answer all your questions, maybe by email after the event. Yes. So Dr. Sosinsky, let's start with you. You work on translating into clinical practice your genome research at the National Health Institute. So I will ask you at the very beginning a general overview on genomic research landscape in England from a clinical perspective and also from a more research perspective. Thank you.
So many young faces in the audience. I was thinking just to put one slide to explain why do we bother about what's the cancer genomics, why do we bother about clinical testing in case of cancer? Cancer can be driven by different environmental factors like smoking. It can be driven by the mutations which are inherited from parents. It can be driven by viruses. You're probably all aware of some cancers driven by the HPV, and also some errors in our genome that we gain during our lifetime. So this is what we do for cancer patients. We collect 2 samples, one from blood, and we expect this to be like a normal reference genome, and also a sample that we collect from tumor. And we doing technique that is called sequencing. We reading these millions of building blocks of the genomes, which we show here with these A, T, C, and G. There are 4 types of them, and they encode, they carry information about proteins that we made of. So every one of these broken pieces of code, substitutions, let's say you change A to G, can be translated into some misfunctional protein. And some of these proteins, we call them oncogenes, that they can drive very quick division of the cells. We call it proliferation, or it can lead to some tumor suppressor genes to be broken. So we also want to pick up those, we call them mutations. So we make this comparison of patient normal genome, patient tumor genome with what we call healthy reference to find all these mutations, all these potential misfunctions in the proteins. And once we know all this information, and we also know clinical information on patient, like what's the diagnosis, where tumor developed, how big it is, we can come up with some personalized treatment suggestions, which hopefully patient will respond much better than just generic therapy. Then, you know, we just treat all triple negative breast cancer with the same platinum therapy. The more we know about cancer is a disease of genome, so more we know about this specific breaks, we better and more efficient can treat patients with more targeted approaches. If we move to the next one. This is how genomic testing is set up in England. I come from company Genomics England. We are a company which is funded by the Department of Health to do whole genome sequencing for patients with cancer and rare disease. And we already have this national network. We have test directory, which tells you what are the genomic testing you're supposed to do for patients with different cancers. Then we have the 7 genomic lab hubs across England that do this sequencing analysis. A more detailed analysis for the whole genome is done at Genomics England. And then once we have the summary of findings, which we think are clinically relevant, we return them to our genomic tumor advisory board to translate them in recommendations for patients. Next one. Yeah, this is the test directory. And it lists some very targeted tests when you just test in 1 gene or 10 genes. And it also tells you that you're supposed to read the whole genome, do whole genome sequencing for few clinical indications. We're doing now for all pediatric patients and also young adults, all neurological tumors, and also all these cases where clinicians think that this knowledge of the whole genome is beneficial for patient treatment. Next. You know, this is why we're doing genome. If you look at the number of tests that you're supposed to do for patients with some indications, it's just very long list, like 24 of tests. You want to know everything about small variants. You want to know whether some genes gain copies, whether some chromosomes are broken, whether patient inherited some pathogenic variants from their parents. So you can do separate tests, or you can just do what we call one-stop shop. You can just read entire genome of the tumor in one test, and this is what we call whole genome sequencing. And you know, this is just probably our most famous showcase. It was all around the media. You can Google for baby Oliver. So this is a newborn baby. He was, well, I wanted to say it because a newborn diagnosed with very aggressive type of sarcoma, infantile fibrosarcoma. And pediatrician already requested to schedule chemotherapy and surgery for this baby. It was a big lump on baby's leg. But they took a bit of the tumor, which called biopsy, and sent it for the whole genome sequencing, and then results arrived. it was just a single mutation in this baby's tumor in the gene called PDGFRB, doesn't matter. And once clinician was aware of this rearrangement in the patient genome, it was very straightforward to change diagnosis to very rare benign tumor called myofibroma. Benign tumor means that it doesn't go into proliferate, so baby don't need any treatment. And chemotherapy in this age can be very damaging. And of course, surgery is also very risky. And actually, in a couple of months, this tumor starts shrinking. So, we save chemotherapy, we save a lot of intervention for this patient with reading patient genome. If you go to the next one, don't worry if you can't read it. I will just explain it very quickly. This is a slide that a colleague from Netherlands shared with me. They're also doing whole genome sequencing for cancer patients. So in all these patients, they identified some mutations, some dysfunctions of the genomes that can be targeted with these drugs which target specific mutation in patient genome. And in some cases, clinicians decided to treat patients with these targeted drugs. And in other cases, clinicians decided to stratify patients for some generic chemotherapy. Like, as I said, a lot of breast cancer, triple negative breast cancer are treated now with platinum therapy without understanding what are the exact mutations that happen in the patient genome. So those patients which were treated with targeted drugs with understanding of genomic rearrangement are shown here in green. And those which were just treated with chemotherapy are shown in yellow. And then, so it was several hundred of patients in green group and yellow group. And then we show in here survival. So half of the patients which were treated with generic chemotherapy unfortunately died at the end of the year. And the follow-up on this patient now is almost 5 years. So for patients who were treated with targeted drug, half of them are still alive after 5 years. So this is the split between green line and yellow line. But you see, it's only work if we give these targeted treatments very early. If it's a second line of treatment or third line of treatment, then the gain is really small. So I think it's very important that I hope within next decades, we will see more of these targeted treatments, which are taken to the first line of treatment with understanding of patient genomics. So this is where we are now. And this is just to show, so most of this was about this left-hand cycle, clinical cycle, when we collect genomic data from genomic material from patient, do whole genome sequencing, analysis, return it back to clinician with expectation that patient management will be improved, but it's not end of the story. We then have flow of data into this research cycle. We enrich it with a lot of clinical data. We know how our patients were treated, whether they responded, how long they survived. So this can feed into this new clinical research design for the new clinical trials. I was on Nordic Precision Medicine conference last week, and I know that in Sweden, due to this collection of data, patients now have early access to drugs. So these drug approvals now is much quicker, but because they have the system of collecting patient response to drugs, they can have this preliminary approval, start treating patients, and 3 years later, they just go back and review how well these treated patients did within 3 years, and then make a decision like, is it a finally approved drug, or, drug is not very efficient, so it's probably not worth to continue with it. But it's very important to collect this data nationally and hopefully internationally across Europe, there is a lot of discussions now about how we can federate data across Europe. There are a lot of rare cancers where you see just a few dozen of patients in a country. So it's very important that we can compare these patients across the borders. So I think I will stop here.
Yeah, yes, you know, in Italy, we haven't yet established a wide national genome sequencing program. So it's very important to say that to our public and to the general public. And in fact, you already show us the benefit, the individual benefit and clinical benefit of this program, but I would like to ask you the long-term benefit for a population of having the genome sequenced. And also if it can promote a more equal access to healthcare systems. This is a very important issue.
Yeah, of course. Well, first, it's very important to start collecting data. And, you know, unfortunately, we have thousands of patients who are diagnosed every year with cancer. So the earlier we start collecting data, the better. If you go to the next slide, no? Sorry. Next one. Yeah, I just pulled here some examples of what we can do next. And you can see that artificial intelligence come on every project now. So we can start looking at more complex biomarkers, not just single mutations in a single cancer driver gene, but in more common signatures. And I will show later an example of cancer of unknown primary. We have few thousand patients every year that come up with diagnosis cancer of unknown primary. That means that patient is presented with metastatic disease, but you don't know where this metastatic disease come from, each organ. So this is probably one of the deadliest cancers because you blindly treating patients with platinum and chemotherapy. So we can think about more precise treatments. Another example, for example, is risk stratification for hematological tumors. So once a patient is diagnosed with leukemia, for example, hematologist is making an assessment. Is this patient under the high risk of recurrence, or you just treat and patient will be cured for next 20 years? And if patients have high risk of developing recurrence, then it's undergo bone marrow transplantation, which very risky operation. So you're making this assessment of risk. And at the moment, it's just a scoring system. It's like, well, I can see this mutation, it gives score of 3. I see this mutation, it gives another like addition. And then you just summarize score, and people frequently do it just on a piece of paper. And in many cases, it's inconclusive. Patients have this intermediate risk. And then it's up to clinician to decide what they want to do. But now we collected a lot of data, which we can use to train AI-based algorithms for this risk stratification. Another example, I spoke so far only about genomes, but of course, genomes and then transcribed into RNA, RNA to the proteins. So we can use these multiple modalities. We also can look at the image data, at patients' clinical data. And we can combine all this information again with AI to predict at the point of diagnosis, is patient likely to respond to chemotherapy? Because this is usually first line of treatment. And in some cases, we can say, oh, this patient is very good responder, and then probably doesn't have to undergo all 6 cycles of the chemotherapy and then be sent for surgery. So we can again save a lot of intervention. Honestly, for me, this cancer genomic is more about treatment reductions, because we still overtreat patients and most of the drugs are quite toxic and surgeries are very expensive. And if we know that patients are not likely to respond to the chemotherapy, maybe we can pool these targeted drugs to the earlier lines of treatment with benefit to the patients. And you probably heard a lot about these personalized cancer vaccines. We have now phase 3 clinical trials from a couple of companies. They're using the same technology that was used for COVID vaccination. And this is tumor-specific epitopes that sit in the surface of the tumor, which are targeted by these cancer vaccines. And of course, it's also used AI methods. I will just go through the top one. If you go to the next slide. Yeah, so this is this case, then patient is presented with metastatic disease and you don't know what was the tissue of origin. And you can extract different features from patient genome, like see which genes are mutated, are there some common patterns of mutations, and feed all this information in some machine learning classifier. I will show on this next slide, like what I am doing today.
Yes.
So this is the case of the cancer of a non-primary. Patient came with metastatic disease in brain. And we find this mutation in, for example, p53 gene, RB1 gene, NF1 gene. Those are pretty common cancer genes. Probably it's a lung, but I'm not sure. p53 is mutated in half of the patients. But then it's also come with very high, we call it mutational burden, very high count of these mutations that we identified in the tumor. And this is characteristics of colorectal lung melanoma tumors. And then most of mutations were these C to A substitutions. Remember these building blocks of your genome. So those mutations we frequently see in smokers in either lung or bladder. So I collected all this information and I can guess that these patients have lung disease. But of course now I don't have to do it just guessing it from my head. We can feed much more features into the AI algorithms. And if you go to the next one, about 70% of patients with these cancers of a non-primary, we can guess what was the tumor of origin for these patients. So they will likely to be treated with more personalized approach. This is not clinically accredited yet. We're still in the process of refining it, but this looks very promising. I always say if I manage to get it into accredited pipeline, I can be retired. So this is how I see future.
A question about the tumor classification you were speaking about. You were showing us the slide and changing our perspective from tumor localization to molecular and genomics characteristics. Do you think we are almost there in this changing?
You know, we have now this very promising study. It started in Netherlands. It was called DROOP clinical trial, drug rediscovery protocol. So for example, if you have drug which targets, let's say, mutations in PIK3CA oncogene, this is a drug, this is a mutation that very frequently happen in breast cancers. And it's already underwent clinical trials. There are a big cohort of patients, so it was approved for use in breast cancers. But we know that these mutations also happen in other types of cancers. So now within the framework of this trial, you can build small cohorts of patients which also have mutation in this PIK3CA gene and treat it with PIK3CA inhibitor. And you first start from treating 8 patients, then grow this cohort to 25 patients. And if you see response, then you can translate it into the proper clinical trial. But frequently, these cohorts of patients very small. For example, BRCA gene in lung patients, you have a handful in each country. But now there are these DROOP-like clinical trials all across the Europe. The study is called now Primrose, and it's kind of umbrella for these clinical studies for each country. And hopefully our cohorts will be growing now much quicker and we can get drug approvals much quicker. And you know, at the end, a lot of the stuff that I discussed today, I hope it will contribute to the equity of patient access to these new treatments. Like, for example, for these cancers of unknown primary, if you are in a big hospital, then you have a big team of oncologists, pathologists who try to guess where your tumor come from, and you know, probably you come up with right answer. If you're in a small hospital, you just go for some generic treatment. Yeah. So I think the more computational methods, like which are come from genomic or multiomic, we can use to make these guesses about patients' personalized treatments, the more equal will be access to the new medicine. And obviously this Drup-like trial across Europe, it's very promising initiative.
Thank you. Thank you, Dr. Sosinsky, for sharing your insights with us. And those advancements are really, really exciting from the patient's point of view also. Now let's go on, and next step or next stop on our journey will be the research below all those results. And I would like to hear from you, Dr. Iorio, because you and your team work on the frontier at the interface, I may say, of biology, statistics, artificial intelligence, and so now I can call you a bioinformatician. So let's get started. What does it mean, bioinformatician?
Yes, so I'll answer this question by asking a question to the audience. So how many of you is a biologist or is studying biology here? Raise your hand. Great. And how many of you is a bioinformatician or is studying computational biology? Well, I suggest those people in the first set to keep their hands in the air, because nowadays, every research, every study in biology is at least at some extent a research in computational biology, right? So in a few words, computational biology or bioinformatics, it's that area of computer science that designs algorithms, methods, and tools to investigate problems in biology or in biomedicine in general, right? But the fun side of the story is that the original meaning of bioinformatics was slightly different from the one that we know today. So this term was coined in 1970s, and the original meaning of this word, according to the people who conceived them, so those were scientists that were interested in studying and understanding how information was processed and would flow into biological systems, like in neurons and then basically the idea, the aims of these people were to implement algorithms that were able to mimic the way information flow in biological system and how basically, for example, our neurons process information. And that's at my back here. It's the first mathematical model of an artificial neuron, right? Which was developed by McCulloch and Pitts as early in 1943, which means that the other fun side of the story is that all the concepts, the basic concepts that are underlying modern artificial intelligence or deep learning neural networks, they were introduced during the 50s or during the 40s in some cases, right? So, but what was missing at the moment, no, that's fine, go ahead. So what was missing at the moment, so basically imagine that it's like we had the design of those models, but what was missing was the engine to make this model run and the fuel, right? So it was like to have the design of a Ferrari, but we were missing the mechanical part to run, to build this Ferrari, and the fuel, right, to make this Ferrari run. The fuel for these algorithms that nowadays are widespread and widely used is big data. And the engine, of course, it's computational power. And so now this is possible because there have been a number of revolutions in technological advancement, and one pertains the surge of a tsunami of data that is available out there and ready to be used to train machine learning models and artificial intelligence methods. And this is just a schematic of how the extent to which the cost, for example, to sequence a genome has dropped in the last 2 decades from 100 millions in 2001, when sequencing the first genome required multiple efforts from multiple institutes and a lot of money, to less than $1,000 that is required now to sequence a single genome. So meaning that, and considering that these advancements have involved many fields, so not only genomics, right? So you can understand now why there is this abundance of big data in biomedicine and in other fields that are making, suitable, this finally suitable, these deep learning methods to be used. And the other, of course, revolution, which is still ongoing and is described by what is called the Moore law, it's our ability in producing, microchips that are increasingly powerful and small and cheap, right? And this law basically shows that every 2 years, the number of transistors that we are able to put on a chip, every 2 years doubles. So, this other basically explosion in computational power paired with the explosion in big data has made now possible the pervasive use of AI in biomedicine and in bioinformatics. And now we are able to, predict with very high accuracy the three-dimensional structure of a protein, or we can make prediction on the way patient would respond to a given drug by looking at its genome, where there is enough data, and so on. So this nicely closed the loop now, right? So we started from methods that are computational methods that were biologically inspired and tried to emulate the way human neurons work. And now these tools are used to solve problems in biology and in biomedicine. So in a nutshell, this is what bioinformatics is, right? So yeah. This is double meaning, and this is basically what we've been talking about until now.
So, sorry. So let's go even deeper. And my next question would be about drug and drug resistance and your big initiative named Cancer Dependency Map Analytics. You have been the leading scientist of the team. Can you describe the method and the goals? Because maybe we come back to that later, but this is a very massive and important initiative from different point of view. So now just goals and the method.
Yeah, so I'd like to start with a dramatic yet powerful cartoon here, which is this one that's shown in this slide, which basically says that globally, worldwide, across all the patients that are presented to the clinic, less than 1/4 of them have a genome that speaks to us and can be used to guide the prescription of a therapy, right? So for the remaining 75% of these patients, there's go back to one slide. Yeah, for the remaining 75% of these patients, there is no other option than chemotherapy, right? Which, as you know, it remains the most effective therapeutic option, the one that works the most, but of course it has a lot of side effects because it tackles biological processes that are widely, extensively used by cancer cells, but are used also by normal cells, right? So we need to do, and then, the situation is even more, serious than this, because even for these patients that can be treated, at a certain point they develop resistance and then they have a relapse of their tumor. But what we do at the moment in my team, we work for this remaining, set of patients for which there is not even a first therapy that is targeted, right? So this is a very crucial problem and something that requires a lot of effort by the biomedical community. The other problem is that even when you have a new drug that is under development, what it happens that when that drug reaches the final phases of clinical trials, the most frequent scenario is this one, right? So the drug doesn't work for a sufficiently large portion of patients because those patients were selected in the clinical trials that in a way that is not genomically informed. So the cohort of enrolled patients is unselected. So they're most often, still today, they are enrolled on the basis of their disease histology only, and the drugs doesn't work for the majority of them, and the trial fails. So, and this happens for 90 to 95% of the cases, which basically explains why the development of new drugs is at a very high critical attrition rate. And why it is vital to work on initiative like the cancer defense map that you mentioned, and which I've had the privilege to be involved since its inception. The goal of these efforts is to identify and catalog all the Achilles heels of cancer, right? So all the weak spots that a cancer cell has, and that can be targeted selectively, sparing basically normal cells, contrary to what chemotherapy does. And yeah, go ahead. So how do we do this? I mean, one way of doing this is by employing a genome editing technology called CRISPR, which was worth the Nobel Prize some years ago to Jennifer Doudna and Emmanuel Charpentier. And I don't know how many of you is familiar with this system, but basically think about CRISPR as a scissor, which can be guided as a GPS, right? So you tell the scissor where to cut. And by designing so-called single guide RNAs that target specific genes, you can tell this scissor to cut wherever you want in the genome. Even further, you can do other things. You can do substitutes of bases or an amino acid. You can change the sequence of a gene, right? So in the projects I've been involved so far, what we've done, we have employed this technology to deactivate one by one all the genes at a genome-wide level in a quite large number of cancer models. And we have measured how these cancer models would react to these gene inactivations, right? And if they were happy or they would die, right? And because these are cancer models that are growing in a lab on a plastic dish, they are fully comprehensively characterized, so we know everything. It is easy now to build predictive models and to that are able to associate to specific responses of this to these knockouts, to this genetic inactivation, associate specific features that then in the future can be used as biomarker. And when there will be a drug that is able to target the coded protein of those genes, can be used to enroll patients in a suitable clinical trial. So in a way, with this initiative, and if you could go ahead on one slide, what we are doing, okay, so it's stuck. So what we do, it's basically, we are not only interested in building predictive models for drugs that are already out there, but we are able to estimate what will be our predictive ability for drugs that do not exist yet, right? And if we are able to predict, in our model, we are able to estimate a high predictive ability for a given genetic knockout, then we use that as a mean to prioritize the development of a drug that then targets the product of that gene. So we are putting together so-called target prioritization strategies that will guide the next generation of anti-cancer targeted therapies.
And about repurposing also is very important to us, because we often say that AI systems see where human brains see nothing. So, but it's very important to see meaningful association. Yep. So what about this and the computational methods you use?
Yeah, drug repurposing was something that Alona briefly mentioned toward the end of her presentation. Repurpose a drug means identify a new therapeutic application for a drug that is already out there on the market, and to prescribe that for a condition that is different from the one the drug was originally designed for. Now, why this is so important and so appealing, especially to drug companies and drug producers? Because as I showed before, the drug development process is at very high attrition rate, and developing a new drug, it's a very long and costly process, which takes up to 30 years. Now, if you have a drug already on the market and you want just to reposition it to another condition, you just need to prove that drug works for the other condition. You don't need to prove that that drug is safe
From the beginning.
because it's already on the market, right? So that means that the drug development pipeline is shrunk from 30 years to just 5 years, right? So, and that's very important. It's very appealing to drug companies because it means, you know, to put on the market a new drug in a very short amount of time, but also it's very important for patients with unmet clinical needs who will have access to a new compound in a much shorter time. Now, this is fun because drug repurposing is what caused me fall in love with biology at the beginning, because when I was graduating in computer science, I was very interested in unsupervised machine learning applied to the processing of audio signals. So we were interested in putting together methods to solve the blind source separation problem. So imagine you have a piece of music, so where there are two instruments playing, how you can teach to a computer algorithm to distinguish what is the piano and what is the voice. So now these are things that you can do very easily with ChatGPT, but at that time, you know, it wasn't that easy. So, but then, you know, my professor said at that time there was a new technology coming out, which were these DNA microarrays. I'm really talking about like 20 years ago, and which were tools through which it was possible to measure the activity of all the genes at once. And there were, you know, this first big data set that were starting to be released on the public domain. And my professor said, why we don't try to apply some of these unsupervised methods to analyze this data, right? And then I started with a PhD in bioinformatics at TGEM, which is the Teleton Institute for Genetics and Medicine. And we ended up with a method that was able to predict new use for existing drugs based on a guilt by association approach. So we were looking at commonalities, undetected commonalities between drugs, and then we used those commonalities to infer a potential new application for an existing drug. So saying in a few words, if two drugs elicit a same transcriptional response, they may share a mode of action, even if they target different intracellular targets, and therefore they may share a therapeutic application, right? So now, you can go ahead with the slide. Yeah, so this is a very simple explanation of the Sonsum ABC model. So this is a model that was introduced by this biochemist, I think it was a biochemist, Sonsum, which basically pertains the semantic links between concepts, between biomedical concepts. And in this cartoon, you can see that there is a drug that is used to treat a disease. But then if that disease shares some similarity with a third disease, then that drug could be repositioned to treat the second disease. Now, imagine if you have a tool that is able to mine the entire body of work that is out there, all the scientific literature that has been published since the beginning of time, you know, so then you can understand why, you know, artificial intelligence might be the most powerful, and why drug repurposing might be one of the most profiting way to be pursued with artificial intelligence.
And be the solution. Yeah, thank you very much. It has been really a pleasure. And it's really exciting also from my point of view. I'm not a scientist, so I can understand everything because they are very clear, but it's really hard to grasp everything. So let's go on and change the perspective. And here it's Cazzaniga with us. Let's change the perspective because now we try to understand all the processes and how do you train and do you train your systems and your models and how you extract a meaningful output. This is the question. Yes, sorry.
Okay, I will copy Francesco, and thank you both for great talks that give a nice way to start. And I will ask a question, okay? So how many of you have heard about GPT or ChatGPT? Fantastic, everyone. And how many of you have heard of AlphaFold 2? Okay, less than half. Very good. So I will start from there. I think it's a story worth to tell because somehow it's something that we get inspired at the Laboratory of Data Engineering at Data Science Park, which I have the luck to coordinate. And somehow what happened there is inspiration for us, for our activities in several projects, especially funded by PNRR, and I will go from there, okay? So AlphaFold 2, in few words, is an artificial intelligence algorithm that learns to solve the protein structure prediction problem. So basically, is an algorithm that can predict from a list of letters, that is the list of amino acids that compose a protein, its three-dimensional structure, okay? So its disposition in space. Why is that important? Well, we've been speaking a lot about genomics. If you go down the way of biology, you will get to RNA and then proteins, and proteins are the responsible of many activities, both in humans and in other beings. And understanding proteins is crucial to understand biology. Now, why is it important to understand structure? It's important because if we understand the structure, we understand more likely the function of protein, and we're more able to create protein eventually that do what we want, or maybe understand what protein that we already know are doing. So typically when we think about artificial intelligence, we think about something that happens very fast, but as Francesco very well said, in fact, things happen much slower than we thought. In the case of AlphaFold, what I want to stress is that AlphaFold stays at least on the shoulders of two big experimental giants. One has been mentioned by Alona extensively, but also by Francesco, which is next generation sequencing. So the ability to cheaply sequence genome of beings, okay, living beings. So we're talking about DNA, but then you can predict from DNA the sequence of proteins, okay? We're able to get many, many sequences of proteins in living beings. So many that we have tons of them. We have, I would say, billions of sequences nowadays, okay? Coming also from very, very far away places like the Arctics and whatsoever. So we have tons of sequences, but the problem is that we want to understand function. And to understand function, we need to pass from structure. Measuring structure of protein instead is extremely hard. X-ray crystallography, which is one of the techniques that you can implement, is a delicate type of experimental assay. You need to crystallize protein and then to pass it through very sophisticated instruments to then do some very complicated analysis in order to understand the structure. It's very expensive, it's very time-consuming, and in fact, we know only few 100,000 of protein structures. So what AlphaFold did is to find a way to bridge the gap between loads of sequences and loads of structure. This was worth a Nobel Prize, but everyone thought that it will kill a subject, the subject of structural biology. Eventually, everyone was thinking that the advent of AI will kill laboratories in a sense. But what happened, if we go to the next slide, is that this Nobel Prize brought, in fact, to have new laboratories. This is the idea. We have AI augmenting laboratories, as Francesco and Alona were mentioning, so that now what is actually happening is that we're able to predict the structure of very, very much more complicated proteins that we were not able to do instrumentally before, or maybe even to predict complexes. And even we're trying to move to predict proteins almost in motion, to understand dynamics, which is getting closer and closer to how proteins function in the living beings, okay? So I want to try to give you just a glimpse of how AlphaFold managed to bridge this gap, which is something that we extensively study in our laboratories, and that takes inspiration also in this case, and I'm happy that Francesco already mentioned, from other fields, from the fields of computational linguistics. In fact, the big question is, how do we make all these protein sequences worth? We have tons of them. How do we make them worth in understanding structure and function? What we do is we copy what we do, in fact, in what every one of you knows, which is ChatGPT. In ChatGPT, more or less, it's not completely true, but bear with me, What you do is you give a piece of a sentence with a missing word. Okay, you're trying to predict the next word, but let's say it's very similar to give a sentence and cancel a word. For instance, here, there is a on a table. Okay, what this model do is to process this data continuously and continuously to understand the context and try to predict what there is in this blank. Okay, so far that this model can predict that with some certain probability we will have book as a good answer, cat as a good answer, but car will be a very, very bad answer. Okay, we'll never have a car on the table. Now, of course, we don't have the feeling for protein sequences as we have for text, but what people did is they did at large scale, okay, exactly the same game. So they started to take protein sequences, which are just sequences of letters, masking some of them and try to predict their outcome, okay? This is what in the backbone of AlphaFold really allowed to leverage sequences to understand and predict better structures, okay? Because if you try to predict missing amino acids, you are modeling very nicely relations between that amino acids that are going to tell you something about their mutual position, okay? But this explains only the two person on the right part of the, what I said up until now explains only the two person on the right side of the Nobel Prize in Chemistry of last year, which are from Google DeepMind that developed AlphaFold 2, that do not explain David Baker, which is instead from Washington University, sorry. The third part of the Nobel Prize, which instead was given for design of new protein. AlphaFold does not design new protein. AlphaFold predicts structures, but these type of models that we are considering can be turned also into designing useful protein. How do you do so? Well, if you mask a large enough part of the protein and you try to predict it, what is going to happen is that you will find a protein that is very likely, okay, very likely to be functional, okay, to exist, in fact, and to fold really in the real life, but you found it completely in silico. This protein is very unlikely to be the same as any other protein that we have been seen before. This is one other perspective of these tools that are called protein language models that we actively study and develop in our laboratory. And perhaps the other very, very interesting application of this that I want to mention, and then I will stop because we need to move forward, is that in fact what you can do is really almost predict, at least locally, okay, for a few iterations, what is the evolution, the possible evolutions of a protein. You know, for instance, all the examples of sequences of protein spikes that have been collected by researchers, and then you can try to guess what are the most likely mutations that will survive of the protein spike that is responsible, for instance, for infecting host of COVID, okay? So if we understand very well what are the next steps, for instance, of viral proteins, we can understand even better how to create, for instance, drugs or vaccines, in this case, vaccines, to prevent them.
Yes, because my question is about the lack of transparency, because the assumption is that you need to have control, and lack of transparency is at the origin of many problems. How do you manage that? And is it still, it is less than a problem today, or is it a problem?
Okay, transparency is definitely a problem, especially when we speak about topics that are very close to treatments and diagnosis, as we've been seeing before. And perhaps the tricky bit of these models is the strategy in which they've been trained itself, okay? So as I was trying to tell you before, so basically this model tried to predict the right word or the right amino acid, okay? But we do not control, okay? It's not a control system where we tell what every single component of this model must do, okay? Francesco showed us neurons, so all of these models are made of billions of those, and eventually they just become what they have to become in order to predict correctly the missing amino acids without us imposing what they have to do. From this point of view, these models seen as an input-output type of construction, they're completely not transparent. They are black box. In fact, very often we hear about neural networks as black box. So if we want transparency, I mean, we need this, eventually we need these models. They're not gonna go away, and we need them on our daily routine, and they're starting to be crucial for clinic decisions. So there's no hope of removing these black boxes. The only thing that we can do, and we want to do as researchers, in fact, is to try to open them. We want to try what is going on inside. Understanding what is going on inside means to try and track computational paths that lead to decision, decode them to try to understand why they make some predictions. Okay? Explainability. This is the subject that works on this is called explainability or interpretability. This is another key aspect that we develop in our laboratory. And eventually, the explainability, what tries to do is to try to explain eventually, in its further extent, what is the functional role now, not of proteins, but of neurons in neural network. Why is that important? It's fundamental for us to understand how they work and how we take decisions. But it's also important because if you understand what a neuron does, you can tweak it. We can intervene. We can modify these models to try to drive them in directions that we think are worth and useful to pursue. So for instance, you might understand that a certain part of this large neural, we're getting to the level of understanding that a certain portion of this very big neural network is having a certain role in shaping or in predicting a certain part of the structure of a protein, so that if we modify it, we can engineer eventually proteins with the functions or at least structure, and then maybe hopefully function that we want. This is, of course, transparency seen in a positive way. I want just to connect maybe to what Alona was saying, and to the fact that eventually these neural networks will be used actually to take decisions on let's say, drugs, let's say, on treatment, okay? So in that case, of course, using these models as black box is even more dangerous, probably, right? Because you will understand that if we take a medical decision, we want to take it for the right reason. We don't want to take a medical decision maybe because there is a bias which is like a prejudice or like a misunderstanding on our condition. We want these models to take that decision because they understood that maybe we have a certain type of disease that needs a certain drug, not because we come from, I don't know, a particularly underrepresented ethnicity or whatsoever. Just to give you an example, and I go back to computer science because also Francesco did it, so I feel free. Imagine there is a very famous example that in the field of images. So you can think about a polar bear, okay? There is a data set made of polar bears on one side and then on bears maybe from Slovenia on the other, okay? And then you train a neural network to try to recognize polar bears from grizzly or from US or from Slovenia, okay? And then eventually this neural network becomes very, very good at doing that. But then people realize that if now you take a polar bear and you put it in a zoo, okay, with a different background, this polar bear is going to be recognized as a grizzly, okay? Because the decision between polar bear and grizzly was not taken for the right motivation. We need to construct models that we can control and we can understand, because otherwise we risk to misuse them, and we want instead to go in the right opposite direction. Okay?
Yes, you already, thank you, really, polar bear and brown bear is fantastic, but you already mentioned the accountability of your systems. And my question is on the outcomes you get and their biological and physical feasibility.
Okay, this is also, I only get tricky questions today, okay?
You're the last one, so.
It's a very good question, but also like a slightly delicate one. I try to make few points on this, okay? Let's go back to AlphaFold because I think it's a very good example, okay? Again, AlphaFold, from sequences, we predict structure, okay? Perhaps the best thing of AlphaFold is not even that it can produce structure, but that it can tell you how much the model is, how much the prediction that the model give you is reliable, okay? So basically, you give me a sequence and I don't output you just a structure, I output a structure and I tell you, yeah, I'm confident on this position, on the geometric properties, let's say 50%, you're not gonna look at that. If I'm confident 90%, you are gonna look at that. This is crucial. This is, we give another name, okay? We said before explainability, this is measure of uncertainty. This is crucial. It's crucial and it's very difficult, but it's one of the most important fields in the subject. Eventually, there are different techniques to measure uncertainty. Some people have Bayesian approaches. Some people, as in the case of AlphaFold, use other artificial intelligence techniques to predict, in fact, uncertainty. But definitely, this word is one word that I want to give as, let's say, a word to think about also for the community of us developer of artificial intelligence, where to move on in the future. Another thing that I think is worth mentioning and bring me back to AlphaFold, okay? At the very first moment, okay, in a bit of a naive way, as a scientist, started to think that, okay, this model is able to predict the structure of a protein. Maybe it has understood the physical properties behind the folding of the protein. In fact, this is like a false hope. We need to also be conscious of the limitation of what we do. I mean, AlphaFold, I can tell, and eventually if I'm wrong, you can shout at me, but I'm very convinced not. AlphaFold did not understand the physical process of folding, okay? What these models understand is different. These models understand co-occurrences between data. They understand relations between amino acids, but they don't understand physical reality. So eventually, this is still ongoing research. There are publications nowadays on what AlphaFold has or has not understood. I think one crucial concept that it's becoming a little bit evident it has understood is this concept of co-evolution, okay? So eventually if you see the same protein in different species, you can understand how mutation influence each other when passing from one species to another. And this is something that this model can learn, can model, because they've seen many, many sequences for many different organisms, okay? But what is crucial is that we are conscious not only of the strength of this model, and maybe we get to the last slide. I have another one because I want to leave it as a closing. Not only of the strength of these models, but also of their limitations. And I connect back to previous talks. There are eventually two approaches. The approach that I try to describe is an approach top-down, okay? We build a very large model and then we try to understand them. There are different approaches that build more physically grounded models that, though very often are not sufficiently, at risk not to be sufficiently accurate. Eventually, probably, one nice perspective on the future is that we have to try to build models that get at the intersection of the two, okay? The next frontier will be of trying to build models which are large and very, very effective, and maybe also with some insight from the physical physical insights that we understood during the years. I leave you with this last slide, which is a snapshot of the protein universe that has been predicted by in silico techniques. These are the goods. We like them very much. There's still, I mean, this is amazing. It's something that we can not have think of a few years ago, but it's not the end because we don't understand much of it. And now comes the nice part of seeing what we can do with these models and with their products.
Thank you, Dr. Cazzaniga. At this point, I would ask the member of our audience if they have any question. The mic there.
Okay, thank you. So I was wondering about AlphaFold. Forgive me if you already answered this question. I'm not really there. I'm a bit sleepy, but you were talking a lot about how we can predict essentially secondary structure of the amino acid sequence, because it really understands well how different amino acids interact and the relationship between them. What I'm wondering about is proteins aren't just made up of amino acids. Most of them are also glycosylated. Many of them have disulfide bridges with tertiary structure essentially. Many of them also exist naturally in some kind of interaction between each other for like quaternary structure of proteins. And my question is how good AlphaFold is at predicting that additional layer of protein structure on top of like amino acids.
Okay, so if I have to be honest, there is only one structure that I didn't show, one layer of the structure that I didn't show on the slides, which is the secondary structure, okay? Because what I show on the slides, if we find them somewhere, okay, there is the primary structure, which is the sequence, okay? The tertiary structure, which is the folded protein. The secondary structure is a little bit in between. I think it's a good question. I don't answer fully, but I add some things here and there, okay? Secondary structure is a fun, I mean, it's great that you talk about that. I'm sorry for everyone if I'm going a little bit too in depth. So in fact, secondary structure is what has been shown you can learn, okay, without having any information of 3D structure, okay? So only by predicting missing amino acids, you are able to recover secondary structure, okay? AlphaFold goes a bit beyond that, so it's not only made of a part that understands secondary structure, building on top of that, resolve the tertiary structure. I mean, basically, the output of AlphaFold is comparable, let's say, on proteins which are not too far away from what has been measured by X-ray crystallography is eventually comparable, okay, with X-ray crystallography. So it does not only resolve secondary structure, but it resolves tertiary structure. And eventually, large part of quaternary structure, I think. But this is what I can say.
Hi. Okay, thanks. I think I'm curious to know your opinion about one thing. In the context of the last 50 years, there's been a lot of technological revolutions. Internet, it came out of a public set of investments. CERN LHC came out as a set of public investments. GPS is something that comes out of public investments. In this specific context, this is the first time that Nobel Prize is awarded to a scientist that works for a private company. And this technology these days, it's completely owned by private companies. Should we be thinking also about the possibility of trying to compete with public sector investments in this direction, or we should just eventually buy the technologies that people will deliver?
Would you go first, Dr. Iorio?
Thanks for the question. So I'll briefly touch on that, and I think that the future it's relies into public-private pre-competitive initiatives, and I think there are examples there, especially in the drug development field, where different companies have joined forces, and they work in a pre-competitive manner, and they say, of course, these are drug companies, so I don't. It's not exactly the field you mentioned, but I say until we discover the target, we work together, and we put everything on the public domain, and then every one of us is free to pursue their own molecule and to get revenues from developing. And I think that's a winning example, and it has brought very quickly to the discovery of new therapeutic targets. And I think maybe it's the same way we should go also for large effort in AI and other fields. What do you think, Alona?
Over to you, Dr. Sosinsky.
Probably my experience comes mainly from sequencing technologies. And I see a lot of interplay between private companies who develop technologies and then public sector who is doing a lot of testing, improvements, provide clinical samples for improving these technologies. So I think at the end, it's win-win situation, because original version of the technology was not quite capable to perform good enough for clinical use. But because clinicians and academic research contributed a lot into, I don't know, developing software that can analyze data, generate a lot of data from clinical samples to test technology, and then feeding back to the private companies which originally developed technology, at the end, we have something that we can offer to the patients. And I think that pharma is changing a lot during, I don't know, last 10 years. I don't know, they probably just made so much money that now ready to give some back. And for example, fund some fancy AI startups without like very obvious financial benefits, but just contributing to the future.
I also say a few things. Actually, I liked the Francesco perspective that I never reflected very much about, probably because I come from a slightly different field, so I will think more about that, and it's great. But okay, today I'm obsessed by AlphaFold, so I will speak about that again to answer the question of Giulio. There is a huge difference between GPT and AlphaFold, not only that all of you know GPT and do not know AlphaFold, but also that GPT is a closed model, while AlphaFold is an open model. I mean, is let's say open weights model. Okay, not completely open, but it's open weights. So as a scientist can look into that, and what I was saying before, looking at neurons, etc., is something we can do, and we cannot do it with GPT. Why is it so? Is because there was a super huge pressure from the scientific community, in fact. The only reason why AlphaFold is open is driven by competition, and institutions, and in particular, groups in collaboration with David Baker, in fact, managed to get the very, very close model that is very, very close in behavior to AlphaFold quite soon. So AlphaFold was put under the pressure of releasing their model. Okay, so eventually, I think the scientific community can have a role in also pushing companies to release their model for the further analysis, and to create also collaboration, public-private from this point of view that can be resourceful.
I hope that this talk has been informative, and now you can ask your question after. They will be more than happy to answer here, but now we run out of time. So I would like to thank you, Area Science Park, for organizing this event, and with those scientists here, so brilliant. I hope maybe next year we can be here together again to hear from your advancements. And thank you very much again. Thank you all for joining us on Saturday night. And we are here happy to answer. Thank you.